Hi everyone, now we're discussing sorbitol, also known as polyol pathway. Very important. Now, what is polyol? It's basically a sugar that comprises of multiple alcohol groups. So, sugar that's also an alcohol is basically polyol. I hope that's clear. So, sugar, also an alcohol, is polyol. So, in this sorbitol pathway, what happens is that we have glucose. That glucose gets converted to sorbitol, and this sorbitol gets converted to fructose. And this fructose is easily usable by seminal vesicles. In seminal vesicles, what do we have? We have sperms, right? So sperms need fructose as a source of energy. So their primary source of energy is fructose. So that's how this pathway is beneficial. So it's that easy, and we're done. Just kidding. Let's revise everything and revise in the sense that we need to know everything in the sense you need to know about the enzymes and also what are the substrates, what is the regulation and all of that. So first of all, in order to convert glucose to sorbitol, you need aldose reductase. Now, let me explain this to you. So glucose, what is it? It's a six carbon carbohydrate, right? And six carbon carbohydrate. Other than that, this is an aldehyde sugar. So it's an aldose, right? An aldehyde sugar. So aldose reductase. So obviously you have to reduce the sugar, aldose sugar. When you reduce aldehyde, so aldehyde reduction produces an alcohol, right? So that's what we are doing. We're producing an alcohol group in the glucose. So you can see this is an additional alcohol group right here. So right here you can see this is an additional alcohol group. So we got the sorbitol. So aldose reductase. We're going to reduce that. Obviously, whenever aldehyde is reduced, you get alcohol. So aldehyde reduction gives you alcohol, and you'll have sorbitol. For this, you also need NADPH. And remember where we created NADPH in the pentose pyrophosphate in the pentose phosphate pathway, right? HMP shunt. So aldehyde it is reduced and you get an additional alcohol or hydroxyl group, and this is polyol. Polyol is basically a sugar alcohol. What is polyol? It's a sugar alcohol. Good. Now what is the okay? After this, you need another enzyme, sorbitol dehydrogenase, which is going to convert it to fructose, and you get a ketone here. Fructose is a ketone basically. So you have your alcohol, you're retaining your alcohol, but you're getting a ketone. So you got fructose. For this, you need NAD positive. So that's it. Sorbitol dehydrogenase, aldose reductase. So aldose reductase and sorbitol dehydrogenase, easy. Now, most sugars, they're rapidly phosphorylated following their entry into the cell. So as soon as they come into the cell, they're phosphorylated. Once anything is phosphorylated, it's trapped. It cannot go outside, and they cannot freely cross the membranes without the need of specific transporters. So um, an alternate mechanism for metabolizing a monosaccharide, this is a monosaccharide, is to convert it into a polyol and by the reduction of an aldehyde group and producing an additional hydroxyl group. So you reduce aldehyde and you produce an additional hydroxyl group. So this sorbitol production basically this will help um, it to easily like now it will get converted to fructose, and fructose is easily capable of leaving your cells, right? Aldose reductase is the enzyme, and other than that, this enzyme is present in retina, lens, kidneys, peripheral nerves, ovaries, and seminal vesicles. But sorbitol dehydrogenase is present in only three things. One is the liver, the other is the ovaries, the other is the seminal vesicles. So seminal vesicles, ovaries, and liver. That's it. Now, this two reaction pathway. So this is a two-step pathway, right? Two-step reaction or two reaction pathway from glucose to fructose. This is of prime importance in seminal vesicles because the sperm cells they're going to use the fructose as a major source of carbohydrate energy. And in liver, what happens is that if this is happen, I told you liver, ovaries, and seminal vesicles. So in liver, what happens is that the sorbitol it is converted into a substrate that can, if we do not have enough, let's say other substrates like we do not have enough of this or we do not have enough of this or we do not need fructose, so what will happen, this will be converted to other substrates which will be used in glycolysis or gluconeogenesis. Now hyperglycemia and sorbitol metabolism. Now insulin is not required for entry of glucose into cells of retina, lens, kidneys, and peripheral nerves. So for them you do not need insulin for getting into the cells. So in those cells during the times of hyperglycemia a lot of glucose enters the cells. That is uncontrolled diabetes mellitus. An elevated intracellular glucose concentration and adequate supply of reduced NADPH. So if there is a lot of glucose in those cells and NADPH. That's a good thing. Now, in those cells, and exactly those were the cells which had aldose reductase as well, they will convert it to sorbitol. Now, increase in sorbitol within the cell, which cannot pass efficiently through the cell membranes and therefore remains trapped in the cell. So, sorbitol also remains trapped in the cell only because this cannot pass through the cell membrane. Now, what happens in excessive hyperglycemia is that this sorbitol dehydrogenase, what if it's busy or it's absent or it's low, sorbitol will start accumulating. And when there's sorbitol accumulation, this has a lot of osmotic tendency and a lot of water will it will come inside. There'll be water influx, there'll be water retention, and this will lead to cell swelling. That's the reason because of osmotic stress in diabetic patients, you'll often see cataract formation, you'll see peripheral neuropathy, microvascular problems, nephropathy, retinopathy, very important. And also, you can see if there is excessive use of NADPH here, so use of NADPH in the aldose reductase reaction decreases the generation of reduced glutathione. So if a lot of it is being used here, so we would not have um, glutathione production, reduced glutathione. If there's no reduced glutathione, we studied in pentose pyrophosphate pathway, pentose phosphate pathway, that reduced glutathione is an important antioxidant because of which there'll be obviously hemolysis also. So that could also be one of the complications of diabetes. So these are the steps. First, for glucose converts to sorbitol, glucose reductase, and cofactor is NADPH. Then sorbitol to fructose, and for that you need sorbitol dehydrogenase and NAD positive. Some tissues lack sorbitol dehydrogenase. There are only three which have it: liver, ovaries, and seminal vesicles. Regulation depends on glucose concentration and aldose reductase activity. So glucose and aldose reductase activity, and there's no hormonal regulation. Its biomedical importance is more of, it doesn't have as such of a biomedical importance, it has more of a physiological importance that fructose is used in sperms. So yeah, pathologically, if there are tissues that lack sorbitol dehydrogenase, all the other cells other than liver, seminal vesicle, and ovaries, in them what will happen? Sorbitol will start accumulating and will cause osmotic damage, and it can lead to cataracts, diabetic retinopathy, neuropathy, nephropathy, right? So yeah.